This is TAU Unbound, the English language podcast of Tel Aviv University. Please welcome your host, Ido Aharoni, Tel Aviv University's graduate, member of the Board of Governors, lecturer, writer, and veteran diplomat.
Welcome to another episode of TAU Unbound. I'm Ido Aharoni, your host, and it is my pleasure to have here in the studio with us Professor Uri Ben-David.
Hi, thanks for having me.
Welcome to our studio, and it's a pleasure to have you. Professor Uri Ben-David is a professor in the Faculty of Medicine at Tel Aviv University and a researcher in the Department of Human Molecular Genetics and Biochemistry. He's also a member of the National Young Academy of Science and is also involved with the Special European Union program in biochemistry, which he will talk about. Welcome again to our studio.
Thank you.
Professor Ben-David, I must say you're a very young professor, and it's a pleasure to have you. Obviously, you're a star, an academic star at Tel Aviv University, and very well known in your area of research. But before you tell us about that, please tell us about your upbringing. I know you grew up in Ramat Gan.
Yes, I was brought up in Ramat Gan.
And how did you discover your attraction to science?
My attraction to science. So in high school, I actually studied chemistry, and I didn't study any biology, but I was already attracted to the exact sciences. And when I think about it now, I think that it's really my military service that got me interested into the scientific methodology, because I served as an intelligence officer. I served for 5 years as an intelligence officer in the Israeli, in the IDF. And, you know, when you gather intelligence, it's actually very similar to how you do science, because you first need to decide to define the gaps. You then need to try to do whatever you can to close these gaps by, gathering information, which is the equivalent of performing experiments. And then you need to analyze the data and basically ask new questions. And so the methodology is actually quite similar to doing science. The big difference is that in intelligence, the whole idea is to be there first and to be the only one that has this information. Whereas in science, it's all about sharing and spreading the information in order to achieve causes that are, for the benefit of the entire humanity. And so after my military service, I thought what I wanted to study at the university. And biology seemed quite interesting, also because it involved many different areas of science. In order to be a biologist, you need to learn some math and some physics and some chemistry and some computer science and so on. So I started, and then I kind of got hooked up on genetics and continued to do a PhD and then a postdoc. And
your PhD was from the Hebrew University?
My PhD was in stem cell biology from the Hebrew University. And then in 2014, I moved to the Broad Institute, which is a joint institute of Harvard and MIT in Boston, to do my postdoc.
So the Broad Institute was established by Eli Broad, who lived in Los Angeles. Actually, I met him back in the day in the 1990s. And if I'm not mistaken, the task of the Broad Institute is to tackle the most difficult subject in medicine, like Alzheimer's and so on and so forth, right? Am I correct?
Right, yeah. So I think that if there are two words that are that I've learned at the Broad Institute, these would be think big. I mean, I think the Broad is all about doing things at large scale, asking important questions, and putting whatever resources that are required in order to address them. They have different programs. One of their program is the cancer program. And my postdoc advisor was Todd Golub. Was back at the time, he was the director of the cancer program. Now he's the director of the Broad Institute. And the idea was, to find big unanswered questions in cancer biology that would also be of practical clinical importance, and try to see how we can leverage the state-of-the-art genomic tools and other types of tools that the Broad Institute is famous for in order to tackle these questions.
So before I ask you this, obviously you brought it up yourself. What are the big unanswered questions in cancer research? But before we go into that, if you can tell us a little bit about the work that you do with the Young Academy of Science and with the European Union program?
So I'm a member of the Israeli Young Academy, which is a group of relatively junior professors from all the universities in Israel across disciplines. And the idea is to go beyond just our own scientific work and try to advance the Israeli academia by large. And this can be, we have projects from helping new recruits or new faculty to basically to get absorbed in the ecosystem of the Israeli academia, all the way to working with youth and, tackling the gender challenge in the STEM fields, and many other projects that are really about taking the some of the best people in the as Israeli academia and try to work together for societal goals.
Very interesting, and the work that you do with the European Union. You mentioned that you
so the European Union. The European Union is, of course, a very important funder of Israeli science, mostly through the ERC mechanism. In biology, they also have what is called the EMBO, which is the European Molecular Biology Organization. They have a Young Investigator program, which is a kind of a selective program for biologists across Europe that meet together and interact and come up with ideas for collaborations. And again, the idea is to advance science across boundaries. And this is very important because all of the work that we do these days, we need the world, we need the support from our colleagues, mostly in the US and in Europe, but of course also across the globe. And so this is a program that, again, its focus is in enhancing collaborations among top biologists in Europe.
Yeah, no question that collaboration is the key in the advancement of knowledge and scientific breakthroughs. Is also it's worth mentioning that Israel is already since 2010 part of two other major scientific initiatives in Europe. One is the Eureka program, and the other is the particle accelerator, the CERN, the CERN. And it's good to hear that you're involved with yet another very important program in biochemistry and biomedicine. So back to the question of what are the big issues that we still need to tackle when it comes to cancer research, and if you can also simplify the various approaches to how to deal with cancer. So, right, if we all agree that the cancer occurs when cells grow out of control, right? This is the description of the phenomenon. Then what are the various approaches to how to tackle this issue?
So these are very, very large questions. I'll try to kind of summarize my view on that. We know now much more about how cancer develops. We know now much more about the genetics of cancer. We also understand that cancer is not just a disease of a single cell or of a cancer cells in the body, but it's actually all about the interaction between the cancer cells and the immune cells and the microenvironment and the organism as a whole. And so one complexity in cancer research is that there are all these different layers, and we need to understand how they connect. It's all about making the right connections, because people tend to focus on what is a relatively narrow field of research, but then the question is how we merge these fields together. But I think the biggest question now is that how to take all the information that we get from the lab and from experiments and from cancer research and translate them into effective cancer therapeutics. Because on one end, there's no doubt that this is the way forward and this is the only way forward. There are some cancer types that were completely a death sentence just a few years ago and are treatable and even curable today. For example, cancer types that are sensitive to immune therapies, which is one of the biggest revolutions of the recent years. And so we're making huge progress on the one hand, but on the other hand, the problem is that sometimes we have a wealth of data, but we're not sure how we can really translate it and treat patients better. And in some cancer types, for example, some types of ovarian cancer, the way that patients are treated today is just like they were treated in the 80s, which is basically we give them poisons, which is what chemotherapies actually are. And so we're trying to bridge this gap between understanding the disease better and then translating it to effective therapies.
Now,
just one point that I think is really important, which adds to the complexity and to the challenge, is that cancer is not a single disease. There will never be a cure for cancer. There will be multiple cures for multiple cancers, because cancer is like an umbrella name for a family of diseases. And each cancer type and subtype has its own evolution and its own etiology and its own biological characteristics, and therefore it will eventually have its own treatment. So,
would you say that we are heading to full and almost total customization, tailor-made treatment for people? In other words, every person would require a different protocol.
Yeah, well, I think it's about hitting the right balance. Eventually, we're definitely heading toward a precision cancer medicine era. And in many ways, we're already there. There are already specific mutations or specific genetic alterations that if a patient has these specific mutations, they will be treated in one way. And if they don't have it, they will be treated in another way. So this is not like science fiction. This is already reality. It's becoming more and more granular, more and more precise. And eventually, I do think that we will have the entire DNA sequence of every human being, and then of every tumor once the tumor is formed, and then we will apply all the biology knowledge that we have, including new AI tools and other tools that are now being developed to tailor the best treatment for each patient. Now, it doesn't mean that each patient would have a different drug or a different therapy. It's just that our ability to fit between, if we have an arsenal of drugs and therapies, and we have a plethora of genetic alterations in cancer, we'll be able to do the matches better. We'll be able to say this patient is more likely to respond to drug X, and that patient is more likely to respond to drug Y, and that will enable us to treat cancer much better.
Now, what is the role of models in this process? Because tell us more about why it's important to understand models in cancer research.
Right. So, what we're really interested in is the actual disease in human beings. The problem is that when you only study actual tumors, you're limited to correlations rather than understanding the cause of, for example, therapy response. And the reason is that every patient is an individual. Every patient has his or her own genomics and his or her own life experience. And so every patient has his or her own tumor, and every patient is treated once. They may be, treated in different with different treatments serially, but they're only treated once. So our ability to infer from a patient's response to an association between, for example, a mutation and the drug response is limited. When we say cancer models, what we mean is a different types of cellular systems where we take, for example, cells from a tumor and we culture them in the lab in a dish, or we transplant them in mice, or we introduce genetic mutations in mice to begin with and wait until they develop cancer. So these are systems that are controllable, and we can use the same cells and, for example, test thousands or tens of thousands of drugs on the same cells coming from the same tumor, or on mice that have the exact same genetic background. So if when we study cancer, we need to combine work that is done in the laboratory, in animal models, in tumor-derived cellular systems like cell lines. Now we have 3D cellular systems that we call organoids because they mimic like actual organs. And we need to combine that with the genomic and clinical analysis of real-world tumors, which are the actual tumors that we get from patients.
And so when you examine those individual tumors and you say that you design a model based on that, what industries benefit from it? So I understand the pharma industry can use that model to test drugs. What else is involved?
Well, the entire world of biomedicine benefits from that, because we use these cell models to study not just cancer, but any disease, and not just diseases, but also very basic biological phenomenon. And we need to remember that almost all important discoveries in biomedicine, you can trace them back to basic biology. So, it's all about understanding the biology deeply. In my case, it's understanding cancer biology. And the clinical trials or the clinical efforts are the last piece in a very, very long chain of events. And it's really important to understand that, because sometimes I think the general public doesn't really understand, why do we need to work with mice, or why do we need to work with simulations, or with why shouldn't we just take all the the funders' money and put it into testing drugs? And the reason is that if we don't do this very basic biology oriented research, we won't be able to develop the drugs, and we won't be able to know who are the patients that will benefit from these drugs. So we need to invest in basic science, and this is also true for cancer biology.
Now, your lab, the Ben-David lab, deals with three different major issues. One of them is called cancer aneuploidy, which I understand is a word that describes the presence of abnormal number of chromosomes in a cancer cell. Tell us a little bit more about that. What is a chromosome, and why an unusually abnormal number is a problem?
Yeah, so this is actually the main field of research in my lab is aneuploidy. And aneuploidy, as you mentioned, is a change in the number of chromosomes in the cells. Now, every normal cell in our body has two copies of the genome, one that we inherit from our biological father and one that we inherit from our biological mother. And these genomes are organized on chromosomes. So a chromosome is basically a chunk of the genome. It's a chunk of the DNA that is surrounded by proteins and basically packaged in the nucleus of every cell. When cells divide, they need to double the number of chromosomes and then segregate them evenly between the daughter cells. And that's the most important principle of cell division. We need to divide our genetic material equally between the two daughter cells. And because this is such a basic challenge for the cells, this is one of the most regulated processes in biology. Now, when this process is deficient, we start to get differences in the number of the chromosomes in the daughter cells. So you can imagine that if we need to have one genome allocated to one daughter cell and one to another, and we have 23 pairs of chromosomes, and there are problems with this segregation, sometimes a daughter cell will get more chromosomes, sometimes the daughter cell will get fewer chromosomes. Now, this phenomenon is mostly known in the general public from Down syndrome, because Down syndrome is an example for aneuploidy. It's an extra chromosome of chromosome 21. And actually, aneuploidy is one of the major issues with normal human development. It's the main cause for a natural miscarriage in the first trimester is aneuploidy. So it's detrimental and when it arises during development. And generally, most normal human cells cannot survive with extra chromosomes or fewer chromosomes. However, in cancer, we have a really interesting paradox, which is that cancers are aneuploid. 90% of the solid tumors and about 3/4 of the blood cancers are highly aneuploid. And on average, over 1/4 of the cancer genome is affected by this copy number change of chromosomes. So we have some chromosomes from which we have more and some that we have fewer from. And this is a really fascinating, first of all, biological conundrum, because how can the cancer cells tolerate it? How can it promote the ability of the cancer cells to survive and to proliferate, to divide, whereas in normal cells it does the exact opposite? So that's the biological curiosity here. But then it also has a huge potential clinical relevance, because this is one of the hallmarks of cancer. As I mentioned, almost all cancers are aneuploid. All of our normal cells in the body are normal, meaning they have a normal number of chromosomes. We call it diploid. So this suggests that if we are able to selectively kill aneuploid cells, then we will be able to selectively kill cancer cells without harming the normal cells in the human body. And at the end of the day, this is the name of the game. This is all what cancer research and cancer treatment is about: how to kill the cancer cells without killing and preferably without harming the normal cells that surround them.
So your research is focusing on how to bring back the balance of chromosomes to its normal level, right?
Not exactly. So what we do, we are trying to ask three big questions. So first of all, we're trying to understand this phenomenon and understand why do we see specific chromosomal changes in specific cancer types? Because when this aneuploidy occurs, then it's not random. We see patterns of aneuploidy in the cancer cells, and we're trying to investigate what underlies these patterns. So that's one question. The other question is to try to pinpoint the most important genes in these recurrent aneuploidies, because by definition, every chromosome has hundreds, sometimes thousands of interesting genes. And then how can we tell the ones that matter from the ones that are just there for the ride? And then perhaps the most important aspect of the work is not to revert the cellular condition to a normal condition, but rather to devise new strategies to kill the aneuploid cells without killing the normal cells. And in order to do that, we have to understand the molecular and the cellular consequences of this phenomenon. So if we understand what type of liabilities or Achilles heels this phenomenon creates in the cancer cells, we would be able to exploit these Achilles heels in order to specifically and selectively target the cancer cells.
Now, how do you kill a chromosome?
So we're not killing a chromosome, we're killing the functional effect of a chromosome. So let me tell you an example from our work. Okay, so we took about 2,000 human cancer cell lines. So these are the models that we discussed before. And we divided them to highly aneuploid. So these are cancer cells that have high degree of aneuploidy, and those that are normal or semi-normal. So they have low degree of aneuploidy. And then what we did a lot of screening. We did genetic screens where we hit every gene, every protein coding gene in the human genome, one after the other. And we did drug screens across thousands of drugs, including most of the FDA approved drugs. And then we asked a simple question. Can we find differences between the response of the aneuploid cells and the response of the normal cells to this genetic or chemical targeting? And we found a specific gene that is called KIF18A, which is an important gene in regulating chromosome segregation. So this is the same process that basically that when it goes off results in aneuploidy. And this KIF18A, turns out that it encodes for a gene, a motor protein gene that is important for chromosome segregation, and it is druggable, so you can actually design drugs against it. And we published this study 3 years ago in Nature, and together with us, 2 other labs also identified this as an important target for chromosomally unstable aneuploid tumors. And it happened to be the case that at the same time, 2 pharma companies were developing drugs against this protein, but they didn't have any biomarker. They didn't know which patients would benefit from this drug. So they basically just developed it because it was known to be a protein that is important for the cell cycle. But then they took our studies and they decided to target their drug to the population of patients that will likely benefit from it the most, which is patients with triple-negative breast cancer and high-grade serous ovarian cancer, which are the most aneuploid chromosomally unstable tumor types. And now there are actually 2 clinical trials that are ongoing with these drugs against these aneuploid cancer types. And we will see whether what we found in a dish with cells, and later others also recapitulated in mice, we will now hopefully soon see whether this holds true in actual human patients. And if it does, then this can be a real breakthrough in treating these types of very, very difficult tumor types.
Well, I'm certainly fascinated by the work that you do. Now, for the benefit of our listeners, that many of them are friends of Tel Aviv University and people that want the university to do well and to succeed, what are the things that you would say are essential for you, the needs that are essential for your lab moving forward, looking into the future?
Well, we combine state-of-the-art experimental and computational tools in order to study for example, what I've described. And this, of course, requires a lot of funding. We need funding for state-of-the-art technologies and instruments. We need funding to recruit the best students and the best postdocs.
So when you speak about state-of-the-art equipment, what kind of equipment, for example?
So, for example, in my lab, we use a lot of sequencing approaches. So both at the single cell and at the tumor level, so DNA sequencing, RNA sequencing, we do proteomics. We also, as we need to look at dividing cells, we need to use very sophisticated microscopes to look at the cells as they divide and when we perform different types of interferences with them. So we have sophisticated microscopes in the lab. We need to work with mice and test some of our findings also in animals, and keeping mice and propagating mice and doing experiments in mice is also expensive. So this is just an example. We also need the computational power in order to run simulations, in order to apply AI to this type of research, which we also do. So there's a lot of money that goes into it. So the first thing that we need is good funding. This funding is not just to directly fund our research, but also we need to be in a strong environment. We need to have good core facilities. We need to have access to equipment that we may not be able to have in our lab, but that we still need to be able to access. And we need interactions with the world. So we need collaborations. We need we need a strong, independent academia in a democratic country in order to keep doing state-of-the-art science.
By the way, in your work, do you have any official relationships with other academic institutions in the world?
Oh, yes. We have many of our studies are close collaborations with many different labs. We have collaborators in many countries, from Australia to India to many countries in Europe, US, Canada. Some of them are really essential for our work. In general, as I said before, science these days is very, we're all very professional, but we all know a lot about a very small size of, like a small part of the puzzle. And we need to team up. We need to collaborate. And the best way to collaborate is not to collaborate just with the people who sit next to you, but actually collaborate with the best people in the world in whatever it is that you need help with. And so in many of our studies, we have, for example, the study that I described before was a collaboration that my lab led, but there were 8 different labs involved from 5 different countries. Okay, in Germany, in Italy, in the US, other labs in Israel. So it's always a team effort, and it really takes a village. It's not something that a single lab can't do that much these days if it doesn't work together. And working together means working together with the world, working together with the leaders in different types of technologies, the leaders in different types of biological questions, in order to ask together an important and interesting question, and then every lab can contribute its expertise, its resources, its angle, even in how it addresses the problem. And this is usually where the breakthroughs come from.
Yeah, I mean, this is inspiring. I always say that, the countries that do well today in the world are the countries that produce knowledge that matters, knowledge that matters, meaning knowledge that is able to change lives and to advance humanity and to design a better future for all of us. And you certainly do that in your own work, and you make us all very proud.
Thank you.
Professor Ben-David, it was such a pleasure to have you here in our studio.
Thank you. It was my pleasure.
Hopefully, we will get to interview you again soon and hear more about your amazing work.
Thanks so much.
This is it for this episode. And to our listeners and viewers at home, goodbye from Tel Aviv until our next episode.